Okay, I hope my screen is visible. Somebody can write in the chat box. Okay, great. So we'll start the class now. Meanwhile, others can join in. So hi, I'm Shrija Ghosh, a PMRF from IIT Kanpur, and I'm taking the problem solving sessions for the course Experimental Biotechnology. Course instructor is Professor Vishal Trivedi from IIT Guwahati, and today is the ninth session in this course series, and we till now we have discussed different biotechnology techniques. We have discussed electrophoresis, we have discussed uh, chromatography techniques. Then we started in the last class, we started with the cell biology techniques. So in the last class, we discussed about different kinds of cells and uh, how do we do cell fractionation. So I'll start with 2-3 questions about cell fractionation, and then we'll move on to the new topic, that is microscopy and flow cytometry. So we'll discuss about different questions related to these topics. So this whole session will be helpful for you to solve your assignment questions for the ninth week. Okay, feel free to ask any question at any point. If you have any kind of doubt, just write in the chat box or just unmute your mic and start speaking. Okay, I'll try to clear as much doubt as possible. Okay. Okay, so let's start now. So as I mentioned, I'll start with 2 or 3 questions of cell fractionation first, and then I'll move on to the microscopy. So starting with the first question, sucrose gradient centrifugation is used to: option A, separate organelles based on size; option B, separate organelles based on density; option C, purify specific proteins; and option D, analyze the cellular composition. Sucrose centrifugation, sucrose gradient centrifugation is used to do what? Option A, separate organelles based on size; separate organelles based on density; purify specific protein; or the last option is analyze the cellular composition. So what do you think the answer would be? Anybody knows the answer can write in the chat box. Okay, we have one answer, separate organelles based on density. Okay, anyone else wants to answer? Okay, so option B is the right answer. So sucrose gradient centrifugation is basically a type of density gradient centrifugation where we use sucrose to make the density gradient. So we can use different concentrations of sucrose to make a gradient in a vessel, and then like higher concentration of sucrose will have a higher density and lower concentration of sucrose will have lower density, and we will have a continuous gradient, and then we will add our samples. So this is how this is done. So our gradient will be formed in this centrifuge tube here. So this is a continuous gradient of sucrose where at the lower part the more denser sucrose solution will be there, and at the top part lighter sucrose solution will be there. That means less concentrate sucrose solution of less concentration will be on the top part, and more concentration will be on the bottom part, and this will be a continuous gradient. Okay, and once uh, we have a question. By lighter sucrose, I mean just the solution concentration. So basically, let's say if I have uh, 0.1 molar sucrose, then 0.2 molar sucrose, 0.5 molar sucrose, and 0.9 molar sucrose, and so on. So 0.9 molar sucrose will obviously have, sucrose is a sugar, right? So 0.9 molar sucrose will have more amount of sucrose, right, in water. Now that means this will have a higher density as compared to the 0.1 molar sucrose. So we will make a continuous gradient. So there are gradient mixers available for making this continuous gradient, where from low to high concentration of sucrose will be poured in this as a continuous mixture. Okay, so the density will be decreasing as we go on top, or increasing as we go from top to bottom. Okay, is that clear? Is that clear, Aditi? So when we add now the sample, which is a mixture, okay, the sample is a mixture of different components, let's say different organelles or anything. It could be a mixture of anything. Okay, now this mixture is added on the top, and now when we are centrifuging this tube, we will do the centrifugation at a particular speed. The speed will be constant in this technique. Now at that speed, when we are letting the samples move downwards, because the centrifugal force will act. So if I rotate something on a fixed axis, if I now rotate something on a fixed axis, what will happen? We will have two types of forces: centripetal force that will be towards the center, and centrifugal force that would be away from the center, right? So centrifugal force will act on these uh, molecules, and they will try to move downwards, right? They are trying to move downwards. Now, since the density is different at each point, what will happen? Because of the density, there will be a viscosity in the solution, right? And higher the density, higher viscosity will be there. Because of that viscosity, friction force will be higher. Like, think very logically, if you have seen any dense solution like honey, okay, all of you must have seen honey. So if I now drop a small ball in honey, and then I drop the same size ball, another ball in a glass of water, obviously the ball that is moving through water will move faster. The ball that is moving through honey would move slower, because honey is so viscous, it will create a friction. So basically the centrifugal force that I drew here, so if I have an object, the centrifugal force will be downwards, but there will be another force which will be due to the friction, right? That would basically resist the movement of the sample. So if the friction is high, the movement of the sample will be slower, and that's how we will have separated samples. So when the sample will reach its own density, so let's say this blue samples, they have a density around this region of the sucrose. So whatever the density of sucrose is there in this region, this blue organelles or whatever the sample was, it has uh, it has the same density. So that's how the separation happens. So they basically they reach to the point where they match the density of the sucrose that is present in the gradient. So basically we separate the organelles based on density. Okay, I hope this question is clear. Moving on to the next question now, question number 2. Differential centrifugation is based on differences in dash of biological particles of different dash. Differential centrifugation is based on the differences in the dash of biological particles of different dash. So you have to fill in the blanks. Option A, size and density. Option B, sedimentation rate. And for the second blank, sizes and density. Option C, for the first blank size, for the second blank structure. And option D, mass and size. What would be the answer? Whoever knows the answer can write in the chat box. Okay, we have one answer, option A, that is size and density. Okay. Anyone else wants to answer? Okay, Aditi says mass and size. Okay. Both of you are unfortunately partially correct and partially wrong. So the correct answer is actually option B, sedimentation rates and sizes and density. So differential centrifugation, so unlike density gradient centrifugation, in differential centrifugation we will have different speeds. Okay, so first we will spin the sample at a low speed, so the particles will be separated based on the differences in their sedimentation rate, and the particles will be having different sizes and densities. So let's say we have a mixture of biological samples. So we can have nucleus, then we can have mitochondria, all the organelles, and then we can have ribosomes and uh, soluble proteins in it. So uh, what will happen when you will do a low speed centrifugation? Only the heaviest uh, particle, that is the one that are having bigger sizes and higher densities, okay, that they will sediment faster. Why? Because the sedimentation rate is faster. Okay, that's separation is based on sedimentation rate. And what we are separating, we are separating samples of different sizes and density. So in differential centrifugation, our goal is to separate different samples of different sizes and densities. In density gradient centrifugation, we are mainly focused on density. So the separation is based solely on density. Okay, but in this case, size and density both will matter for a particle, and the principle behind the centrifugation remains the same. Any kind of centrifugation, the principle is sedimentation rate. Okay, the separation, like centrifugation, will always depend on the sedimentation rate of a particle. Okay. uh, while sedimenting, which quality is preferred, size or density? Depends on what technique you are using. So if you are using density gradient centrifugation, then we are exploiting only the density, like the property that we are exploiting there is density. Okay, so we are using density as the factor to separate molecules. Now, if I am using differential centrifugation, I am using the property size and density both. It's like depends on what like how you are just, if you are just centrifuging to remove some particulate matter from a solution, you will just take the tube and centrifuge it in a like at a constant speed for some 5-10 minutes till the particles settle down, right? So in that case, particles are settling because both the particles have a higher size and higher density. Like it's a mixture of both. Okay, but depends on, if you are doing a density gradient centrifugation, in that case size actually doesn't affect that much, because the separation is solely based on density. Size will affect because if the size is bigger anyway, a lot of times in cellular components it is more denser also, but it's not always the case. You can have a vesicle, let's say. So vesicle could be bigger in size, but since it's hollow from inside, it's just a membrane, vesicle membrane bulbed which has blebbed out of the cell, so that can have a higher size as compared to other organelles, but it would be lighter. It would be having a lesser density than the other organelles. So in that case, when you are doing differential centrifugation, for example, it will depend on both size and density combined together, and then only we can see, like when we'll do the experiment, we can see what is settling faster. So in each case, one property will kind of dominate over the other. Is that clear? Okay, great. So this question is also clear, I hope. I'll move to the next question. Question number 3. Which of the following statements about the basic principle of sedimentation is false? Option A, denser a biological structure is, the faster it sediments in a centrifugal field. Option B, the more massive a biological particle is, the slower it moves in a centrifugal field. Option C, the denser the buffer system is, the slower the particle will move in a centrifugal field. And option D, the greater the centrifugal force is, the faster the particle sediments. Which of the following statements about the basic principle of sedimentation is false? Option A is denser a biological structure is, faster it sediments in a centrifugal field. You have to select the false statement, okay? More massive a biological particle is, the slower it moves in a centrifugal field. The denser the buffer system is, slower the particle moves in a centrifugal field. And last option is the greater the centrifugal force is, faster the particle sediments. Okay, Aditi says option B. More massive a biological particle is, slower it moves in a centrifugal field. Okay. Anyone else wants to answer? So option B is the correct answer. You are right. More massive a biological particle is, the slower it moves. It's a false statement, because more massive the biological particle is, it will actually move faster in the centrifugal field. So I told you that uh, centrifugal force is acting on the particle, and the denser, the bigger particle it will be, it will have a higher sedimentation rate, and it will have like experience a higher centrifugal force. So the denser the biological structure, faster it sediments. That is correct. More massive biological particle is, it will move faster in the centrifugal field. So this statement is false. Denser the buffer system, slower the particle move. I just explained how something will move slowly in honey, but the same thing will move faster in water, because honey is denser. So any dense buffer system, the sample will move slowly. Then the fourth option is, the greater the centrifugal force is, the faster the particle sediments. That is also correct. If you apply more force, you are technically pulling the sample down faster. Okay, I hope this is clear. Now I'll move to the next question. From now, we'll start the microscopy related questions. So question number 4. Which type of microscope is better suited for observing thick specimens like tissue culture flask or living cells in a culture dishes? Option A, upright microscope. Option B, inverted microscope. Option C, both are equally suitable. Or option D, neither can be used for such specimen. Which type of microscope is better suited for observing thick specimens like tissue culture flask or living cells in a culture dish? So Rosie has answered inverted microscope. Okay. Anyone else wants to answer? Nobody wants to answer. Either you agree with Rosie saying option B is correct, or you don't know the answer. Okay. Okay, it is option B, uh, inverted microscope. We have, like, in the category of light microscope, we have two types of microscope, upright and inverted. Okay. The major difference between upright and inverted microscope, the whole principle behind both the microscope is same. You will have a light source that will illuminate your sample, and then there will be different lenses that will magnify the sample, and you can look into it through a eyepiece. The difference between the upright and the inverted microscope is, in the upright microscope, this is the stage where you are keeping your sample. Okay. Now, in case of upright microscope, you can actually move the stage up and down. The objective lens is fixed here. Okay. So this is the objective lens, right? So this is fixed here, and we can move this stage up and down. The stage can go up and stage can go down. So we'll keep our sample here like small slide or something, and this is the knob. So this is basically how we focus. So when you rotate this knob, so this will either move up or down. So if I want to observe a sample closely, what I'll do, I'll move it up so that it reaches uh, almost near to the lens, and then I can view by looking into this eyepiece, and I can see what the samples are looking like in a microscope, in a magnified field. Okay, so this is an upright microscope. Now, as I'm saying that this stage is moving up, it will be very close to the lens, right? So I cannot keep a whole petri dish on this sample, right? I can keep only thin slides with the samples, uh, like with a bacterial or a fungal sample. I cannot keep a whole petri plate or a culture plate on this, because that will be very big. It will be a very big dish, right? So we don't have enough space between the lens and the stage where we are keeping our specimen. Now, for that, this inverted microscope was developed. What was the difference in this? The whole principle remains the same. So this is the light source in the upright microscope, where the light is illuminating the sample that is kept on the stage, and then we are looking from above. Here, the light source is present above, so it will illuminate the sample from top. We'll keep our sample. So here the stage is fixed, like we had the objective lens fixed here, we had fixed stage in case of inverted microscope. That is the only difference. And we can move this objective lens up and down. So this lens can go up and down by moving this, rotating this knob. This is the focus knob. So when I rotate this knob, this lens can uh, go and almost near to the stage and magnify the uh, magnify the sample, and this is actually connected. So when we are looking from through the eyepiece, we can see what the magnification is happening here. Okay, so here now the stage is fixed, and we have a lot of distance between this area, right? So, and we are not moving the stage up and down, so there's no space constriction. So that's how we can keep bigger plates, like petri dishes or big culture plates, or let's say 6 well plate or 96 well plates. Those things we can keep here easily and look from the look from the top part. So mostly inverted microscopes are used in case of mammalian cell culture, whereas upright microscopes are used in case of bacterial or microscopic microorganisms that we have. So generally bacterial sample, or if we are viewing a fungal hyphae, so we can just pick a little bit of fungus from the plate using forceps, keep it on a glass slide and cover it with a cover slip, put some drops of uh, hydrating, any hydrating buffer or something, and then put a cover slip avoiding the air bubbles, and then keep your slide here and observe through an upright microscope. Or for bacterial sample, you can fix the sample on the slide and then observe through a upright microscope using appropriate stains. For inverted microscope, we generally use it in mammalian cell culture, where we directly keep the culture plate, so we don't have to actually take the sample out and then put it on a glass slide. So we are actually saving ourselves from taking so much effort. We can directly just take the plate out of the uh, incubator and directly keep it under the microscope and see how many, like how our cells are looking. If you add a fluorescence light here, like fluorescent source here, you can actually see fluorescence also in that same sample. So those kind of things. So inverted microscope is more useful in case of mammalian or insect cell culture. Okay. I hope this makes sense now. Moving on to the next question. Question number 5. Which part of the compound microscope helps in gathering and focusing light rays on the specimen to be viewed? Okay, so I just showed you the diagram of the microscope. I don't know if you saw it very keenly or not. So let's see if you can answer this. Which part of the compound microscope helps in gathering and focusing light rays on the specimen to be viewed? Option A, eyepiece lens. Option B, objective lens. Option C, condenser lens. And option D, magnifying lens. Anybody knows the answer? Okay, two people are saying option C. So that is the correct answer. Option C, that is the condenser lens. So in this diagram, you can see here we have the condenser lens. So basically, whatever the light source that is there to illuminate the sample, the condenser will gather all the light and illuminate the sample. In this case, in case of inverted microscope, we have the same thing on the top, because we are now illuminating the sample from the top. Here we are illuminating the sample from the bottom. Okay, so all of you answered it correctly, so I'm hoping that you understand this part. So this is the basic of microscopy, and then based on this, people develop different other techniques of microscopy, where they change the light source, or they change the uh, lenses, different like to have a better and better magnification, to have a better resolution of the images. But this was the most basic design of the microscope, where you can actually observe almost all kinds of cells. Okay, moving on to the next question. Question number 6. What is the primary role of the dichroic mirror in an epifluorescence microscope? What is the primary role of the dichroic mirror in an epifluorescence microscope? Option A, to block the emitted light. Option B, to allow the excitation light to pass through the specimen. Option C, to direct the emitted light towards the detector. And option D, to adjust the magnification. What is the role of the dichroic mirror in an epifluorescence microscope? to block the emitted light, to allow the excitation light to pass through the specimen, to direct the emitted light towards the detector, or to adjust the magnification. Okay, Saugat has answered option B, to allow the excitation light to pass through the specimen. Okay, anyone else wants to answer? Okay, so you are right actually. Option B is the correct answer. Dichroic mirror is added to the fluorescence microscope to allow the excitation light to pass through the specimen. What does it mean? So in a fluorescence microscope, so principle of fluorescence is basically you if you excite a sample with a particular wavelength, and if it shows fluorescence, what will it do? It will emit a higher wavelength. Okay, that is the basic principle. So whatever your excitation wavelength is, that will be uh, like that will excite the sample, and the sample will emit a higher wavelength, and that we see as the fluorescence. Okay, so here this is the simple ray diagram of that. So we have a light source. So let's say I want to excite this sample here, and I'm so I'll use a excitation filter. Excitation filter is basically something that can filter a particular wavelength out. So that particular wavelength now will reach here. This is the dichroic mirror. That it will hit the dichroic mirror and get reflected. Okay, it's a mirror. It will reflect things. It will reflect the light beam. The light beam gets reflected, falls on the sample. Now the sample is fluorescent. The sample will emit a higher wavelength light. Now that higher wavelength light, that is this red colored light, this red colored light is having higher wavelength. So it can actually pass through this dichroic mirror. So property of dichroic mirror is it can reflect lower wavelength lights, but it can actually uh, the higher wavelength lights can actually pass through the mirror. Okay, so excitation light will pass through the specimen. So the excitation light that is falling, excitation light will hit the sample, and whatever is emitted actually, so it should be technically it should be emitted light passes through the dichroic mirror. The excitation light will always reflect, and then emission light would uh, pass through the dichroic mirror. Okay, so but it's the same light source, that's why they have named it like that. Why does the sample emit a higher wavelength in the first place? So it's a property of fluorescence. So if you read about fluorescence, you will see that any sample that exhibits fluorescence, they can actually uh, emit a higher wavelength of light when excited by a particular wavelength. So if it's a green fluorescent uh, sample, if you emit it with blue light, it can actually emit if you excite it with blue light, it can emit a green light. Okay, so that is a principle behind fluorescence. I can't explain this in detail because it would be beyond the scope of the subject. But this is a property of fluorescence actually. When you excite a sample with a particular wavelength, so whatever the wavelength would be, depending on the fluorescence property that is there in the sample, it will emit that light at a higher wavelength. It will get excited basically. Okay, so it is going to an excited state. Whatever initial wavelength we are giving, that wavelength is getting excited, it is reaching to a higher value, and then it is emitted out of the sample. So that is what we see. Okay, so that's a dichroic mirror is used, so that one type of wavelength will reflect but another type of wavelength will pass. So once the wavelength passes through the dichroic mirror, it can directly reach the emission filter or the whatever detector camera will be there. It can reach the detector and we can see the photograph. Okay. Moving on to the next question, question number 7. Dash is used to fix dash is used for fixing cells for immune localization experiments. Option A 1% Triton X100, option B BSA, option C 1x PBS, option D methanol and acetone. Dash is used for fixing cells and for immune localization experiments. 1% Triton X100, option B is BSA, option C 1x PBS, and option D methanol and acetone. Okay, you're saying option A 1% Triton X100. Okay, anyone else wants to answer? Nobody? So for fixing cells, what do we mean by fixing cells? So when we are doing microscopy experiments, uh, we generally fix the biological sample so that everything is like fixed in its position. Okay, so if you take a live cell, so everything is going to move. It's a biological sample, right? It has cytosol. Things are not very solid inside a cell. Things are very fluid. Things are moving even inside the membrane. Membrane is also a fluidic model, right? It has a fluid mosaic structure, if you remember studying this ever. So everything is very fluidic in the biological system. So when we are doing an imaging, if I want to capture a picture, think of it in this way. If I'm capturing a picture, if I'm taking a photo of a person and the person keeps moving here and there, can I get a stable photo? No, right? So I need to fix that person first in a particular position, ask him or her to pose, and then I'll click a photo. So same thing, I have to fix the cells so that they are not moving here and there. The things inside the cell are not moving here and there, and then I can take a picture of that. So for that purpose, we fix the cell. So we can't use Triton X100. Triton X100 is basically a non-ionic detergent. It is mainly used for membrane solubilization. So if you want to break down the membrane, solubilize the membrane lipids, generally we use 1% Triton X100. BSA is a protein. It's bovine serum albumin. It's the albumin protein extracted from the serum of a bovine. And then 1x PBS is just a buffer. We can do fixing using methanol and acetone. So the correct answer is methanol and acetone. So immune localization experiments is actually experiment where we can fix the cells and then we can add some antibody specific for a particular, let's say, particular organelles, antibody specific for a mitochondrial protein. So now we can see if that mitochondrial protein is indeed inside the mitochondria, or if we want to see where the particular protein is localized. So we can take an antibody against that protein and then tag the protein with, like, tag the fixed cells with the antibody, and we can see wherever the antibody is bound, that means that is the location where our protein is localized inside a cell. So to view all these things, we need to first fix the cells, because if things are moving here and there, it would be very hard for us to figure out where the uh, particular thing that we are trying to look at, where it is present inside the cell. Okay. 1x PBS is buffer. It's a buffer. It's a phosphate buffer saline. PBS stands for phosphate buffer saline. So it will not do anything. Generally, it's used for washing the cells. For washing steps, we don't wash things with water because there will be a difference in osmolarity. So phosphate buffer saline has phosphate salts, sodium phosphate, potassium phosphate, and it has sodium chloride. So it kind of maintains the pH and the osmolarity of a cell or any biological material. Okay. Cell tag with antibody. Yes, so that is what I was talking about. So that is how we do the immune localization experiments. Okay. I hope this is clear. So moving to the next question, why is fixation of specimen an important step in immunolocalization? So I just now explained, so I hope all of you should be able to answer this. Why is fixation of the specimen an important step? Option A, it enhances the specificity of a primary antibody. Option B, it preserves the cellular structure and prevents degradation of the proteins. Option C, it amplifies the signal from secondary antibodies. And option D, it is not necessary in immunolocalization protocol. So tell me why fixing is important. I just now explained so much, so if you have understood, you should be able to answer it, all three of you. Why fixing is important? I just now told you what is happening during fixation. Why am I fixing it? Aditi says option A. Okay, that means you haven't understood the previous question. Anyone else wants to answer? uh, all of you haven't understood the fixation part. Okay, don't you think option B should be the answer? I just now explained we are fixing the things. We are basically fix preserving a structure, and then we are looking where. So specificity of primary antibody will have nothing to do with the fixation step. We are doing the fixation step so that we can let the antibodies bind to its antigen easily, but the binding specificity is not going to be enhanced or destroyed by the fixation step. It would just be easier for the primary antibodies to bind to its target. Fixation is mainly done to preserve the structure. I just told you that I need to fix everything. I need to make it a static system. Then only I can let something go and bind to that system. Right? I told you that if I want to click a photo of a person, I need to ask the person to stand still first, right? So that is what I can't ask the cell to be still or I can't ask the things inside the cell to be still. So I have to add things to make it still. So generally methanol and acetone solution is added to fix the cells, to preserve the cell structure. Once the cell is fixed, like nothing is moving inside the cell, now we can add the primary antibodies and then the secondary antibody and do the imaging. Okay, take the photograph of it. Okay, it also prevents protein degradation because the reagents that we use for fixation basically kind of cross links with the proteins. So our proteins are, so it's like some you have just immobilized everything. It's not moving now, so nothing is going to degrade. There's no movement happening anyway, so there's no point of degradation anyway. Okay, so correct answer is option B. So the fixing is the first step, and it is required mainly for two purposes. First, to stop all the biological activities. As I told you, like things are moving inside, enzymatic reactions are going on. So cell is a very active biomaterial, right? Inside the cell, a lot of dynamic processes are going on all the time. So we have, if we want to capture a picture out of it, so we have to first stop everything that is happening, stop all the movement that is happening, and then we can proceed. So it stops the biological activity and it stops the relative movement of the cellular components and intracellular macromolecules. In addition, it reduces the damage to the cellular system and morphology because now the things are fixed, so get things cannot get damaged easily. And generally fixing is done with methanol and acetone solutions. 7:3 mixture is made, and it is done at minus 20 degrees Celsius at a freezing point, like at a lower temperature basically, for 15 minutes. Then hydrate the sample with 1x PBS. So I told you PBS is a buffer. So once you add PBS, the cells are kind of now in its own osmolarity. So if we don't do this hydration step, so after fixing it, if we directly take the samples out, they can burst because now the samples are kind of, uh, you can say they are kind of immobilized, and they are kind of as a frozen. It's not frozen by decreasing the temperature, but it's kind of now static. So you cannot immediately expose it. You have to hydrate the sample with 1x PBS or buffered saline, so that it comes to its like pH, so it will get its own pH and osmolarity, so it won't burst or it won't get destroyed. Is that clear? Both of you answered option A. I was surprised. I thought you would be able to answer this. I hope that's clear now. Okay, great. So moving on to the next question, question number 9. In the immunolocalization protocol, what do primary antibodies specifically bind to? Option A, the specimen. Option B, the fluorophore. Option C, the secondary antibodies. And option D, the target antigen or molecule. In the immunolocalization protocol, what do primary antibodies specifically bind to? Primary antibodies specifically bind to. Option A, specimen. Option B, the fluorophore. Option C, the secondary antibodies. And option D, the target antigen or molecule. Yeah, Rosie has answered option D. Anyone else wants to answer? Okay. Saugata has answered option A. Okay, I think there's still problem in understanding. So Rosie is actually right. So primary antibodies bind to the target antigen or the molecule. So if you like in the uh, immunostaining protocol, we discussed about Western blotting and techniques like that, right? So in that also the same principle is used here also. So let's say we have a cell, okay, we have a cell here, and inside the cell we have mitochondria, and then we have a nucleus, and we have other things happening here and there. Okay, so now first I have done the fixation, so I have fixed the cells. Then after fixation I'll do some blocking steps to prevent non-specific binding, and then I'll add my primary antibody. So primary antibody basically will be specific to whatever I want to see. So let's say I want to see a protein if it is present inside the nucleus or present inside the mitochondria. Okay, so I know there is a protein, so I'll generate antibodies against that protein, and I know that that protein either goes to nucleus or mitochondria. Okay, but I don't know where it is actually going. So I'll add the primary antibody. That primary antibody will now, so in the last class I have also shown the primary like an antibody structure. So a primary antibody will bind to that particular protein against which I have generated the antibody. Now to this I'll add a secondary antibody. After addition of primary antibody, I'll add a secondary antibody that will have some enzyme or some fluorophore link. So that secondary antibody will be specific to this primary antibody. So that specific antibody will now come and bind to this primary antibody. And this has some kind of fluorophore or a detection uh, method that will light up, and I can see, so wherever I can see the signal, wherever I can see the fluorescent signal, let's say I'm seeing the fluorescent signal inside the mitochondria, but there's no fluorescent signal inside the nucleus. So that's how I can understand that okay, this protein is going into the mitochondria and not the nucleus. So that is how this kind of experiment, immunolocalization, is done. Okay, so primary antibody is always specific to the target molecule or target antigen, whatever if we are targeting a protein, then it would be specific to that protein. If we are targeting an organelle, it would be specific for that organelle, and so on. And secondary antibody will be specific to the primary antibody. Okay, is this clear? So Agata, you have answered option A. Aditi says, ma'am, did I miss something? Well, I got it. No, no, no. This is the next question we were discussing, Aditi. Okay. Okay, so all I hope all of you understood this. So I can move to the next question now. So the correct answer for this question is option D, target antigen or molecule. And move to the next question, question number 10. What is the purpose of blocking in an immunolocalization protocol? Option A, to permanently attach the specimen to the slide. Option B, to prevent non-specific binding of the antibodies. Option C, to increase the fluorescence intensity. And option D, to amplify the signal from the primary antibody. Why are we doing the blocking step in immunolocalization protocol? To permanently attach the specimen to the slide, to prevent non-specific binding of antibodies, to increase fluorescence intensity, or to amplify the signal from primary antibody. Aditi says option B, to prevent non-specific binding of the antibodies. Okay. Anyone else wants to answer? Okay, we have one more option B. So option B is the correct answer. We do the blocking step to prevent non-specific binding of antibodies. So as I was explaining in the previous slide also, we do the fixation and then we do blocking. So blocking basically is done using BSA, bovine serum albumin. So BSA will go and bind to all the surface. So then when after addition of BSA, when we add our antibody, it will only bind to the target molecule for which it will have higher affinity. It won't go and stick to other locations because those locations are not blocked. Okay, so blocking is done for preventing non-specific binding of the antibodies, non-specific binding of the primary antibodies. So I hope this is clear. I'll move to the next question. Question number 11. What would be the maximum resolution that can be achieved if a sample is illuminated by green light of 514 nanometer and observed through an oil immersion uh, and observed through an oil immersion objective with a numerical aperture of 1.6. So what would be the maximum resolution achieved by illuminating a sample of a wavelength of 514 nanometer and objective of numerical aperture of 1.6? So it's a numerical based question. I'm not expecting you to solve it right away, but I can ask you one thing. Any one of you can tell me what resolution is. If you know what resolution is. Do you know what is resolution? Okay, I am assuming you don't know. Since you are not answering, I am assuming you don't know. Then, so resolution in very general terms, I'll explain first. So, if I'm again, if I'm taking a picture, so all of you are using phone nowadays, so you must be aware of the term resolution. We usually use it also in our day-to-day life when we are talking about photographs and we say this photo doesn't have good resolution, or it looks blurry, it's not very clear picture. So all these terms are actually it will make sense mathematically also. So basically uh, we also use the term pixelated, that the photo is pixelated. When we are zooming it, it's getting pixelated. So basically when you are magnifying it, it has up to certain limit only you can see two things separated. Okay, so resolution in very simple terms is the minimum distance that can be seen between 2 points. Okay, and in most scientific way also, this is the definition for resolution. So if I've clicked the photograph and now I'm zooming in it into the photograph, after a point I cannot see 2 points very distinctly. So if I've clicked a photo of a person standing beside a tree, I I keep on zooming into the picture, after a point I won't be able to differentiate between the two leaves that are very far away and that are in the picture that are very close to each other. So my photo will not be able to resolve between those two leaves separately. It won't be able to identify the two leaves as separate entity. It will look like a big green blob. Okay, so that is basically resolution. So here also in microscopy is basically imaging technique, right? So whatever applies to a photograph that you are clicking with your camera, the same things are, can be applied in case of microscopy also. It's just that in case of microscopy you are taking the image of at a microscopic level using uh, using the lenses of higher magnification. You are magnifying the samples, you are magnifying the very small minute microscopic samples that you cannot see by naked eyes, and you are using a larger lenses basically of higher magnification to look at it. Okay, so that is all microscopy is. So resolution also, the definition of resolution remains same for microscopy as well. The ability of the microscope or the ability of the lens to distinguish between the 2 points. So the minimum distance it can distinguish between 2 points. So let's say I have 2 points, one here and one here. Okay, distance between these 2 points is 100 nanometer. Okay, let's say my the microscope that I have, the resolution of that microscope is uh, let's say 70 nanometer. Okay, so that means it can see the particles that are uh, separated by 70 nanometer or more. So these particles are separated 100 nanometers apart. So I can clearly see these two particles as two separate entities. Okay, so let's color one particle with green. So I can differentiate between the green and the red particle very clearly. Okay, but when we have a particle separated, let's say we have a red particle here and a blue particle here, which is separated by 50 nanometer now. Okay, then in that case what will happen, since my resolution is higher, I won't be able to see these two as separate points. I'll see both of them merged together. Okay, I won't be able to distinguish between the red point and the blue point separately. Okay, so that is the definition of resolution. Now these values that are given, so resolution is a function of wavelength and numerical aperture. So it is directly proportional to the wavelength and inversely proportional to the numerical aperture. Okay, so the formula for resolution here is d is equal to lambda divided by 2 times numerical aperture. Lambda is the wavelength. So the term resolution is the minimum distance at which two distinct points of the specimen can still be seen. Okay, so your eyes are also a type of lens, right? You have a lens in your eyes and you are seeing things through it. So the same optics principle is there behind your eye. So when you are looking at an object far away, so there is a limit till which your eyes can actually distinguish between 2 points. Like it can actually see like if you are looking in a at a uh, clear field and you are looking at a tree that is far away, you can, your eyes will have a limit till which it can see two trees separately, or it is seeing only one big green merged thing. Okay, so that is the resolution. Resolution of a microscope is linked to the numerical aperture as well as the wavelength of the light used to examine the specimen, as I have mentioned. So this is the formula that is used. So now we have the values. So solving this question is very easy. You have the values, you have the formula, just put it in the formula and you can get the answer. So D is equal to lambda divided by 2 times numerical aperture. So lambda is given as 514 nanometer and numerical aperture is given as 1.6. If you solve this, you will get the resolution equivalent to 160.6 nanometer. That is option number C here. Okay, so that means this microscope is able to uh, distinguish between 2 points that are spaced apart by 160.6 nanometer or above. Below that, it won't be able to uh, dif differentiate between 2 points. Okay, is this clear? Is the concept of resolution clear to everyone? Because that is more important. Solving this type of question you can do if you know the formula. You just have to divide the numerical aperture and wavelength. Okay, great. So this was a direct question where you had the wavelength, you had the numerical aperture, and you can directly calculate the resolution. But then if there is a situation based question, then you should understand the concept of resolution first before answering any such question. Okay, so for this question the correct answer is option C, as I have mentioned. Moving on to the next question now. Question number 12. Dash is used to remove water from biological samples before SEM or TEM. SEM is scanning electron microscopy and TEM is transmission electron microscopy. Dash is used to remove water from the biological samples before doing SEM or TEM analysis. Option A, washing with gradient solution of ethanol. Option B, 5% glutaraldehyde. Option C, overnight soak with 1% osmium tetroxide. And option D, air dried in a desiccator. How do you remove water sample or like water contamination from a biological samples? How to remove water from biological samples? Okay, Swagata has answered option A, washing with gradient solution of ethanol. Anyone else wants to answer? So option A is the correct answer here. So option A, that is washing with gradient solution of ethanol, it is done to remove the water from the biological sample. So why do we have to remove water? First of all, uh, because when we are doing electron microscopy, the water can actually get uh, like water can form gas, like it can get outgassed and it can the gaseous particles released from the water can actually interfere and scatter the electron beam. So that's why water has to be removed. For removing water, we generally wash it with ethanol. So this is the process how we do the dehydration step where we are removing the water. So ethanol is a hygroscopic molecule. So it can absorb water. So but we cannot directly add 100% ethanol to the sample. We need to slowly increase the concentration of ethanol. So basically we have to give a gradient concentration of ethanol. So biological samples are fragile and contains large amount of water. Water present in the biological sample diffract electron rays and may increase the background signal, as I've already told you. So following the fixation step, the osmium fixation step, water is chemically extracted from the specimen using a graded series of ethanol. It is performed in the following steps. The steps are where you are incubating the sample first with 50% ethanol for 30 minutes, then you incubate the same sample with 70% ethanol. So you are increasing the concentration of ethanol. Then you incubate the sample for 30 minutes in 80% of ethanol, then you incubate the sample in 90% of ethanol for 30 minutes, then 95% of ethanol for 30 minutes, and then you go for 100% ethanol for 30 minutes, and then finally incubate it in anhydrous ethanol, that is completely dried ethanol, ethanol free from any water. uh, that anhydrous ethanol will be used for uh, incubation at the final step, and that anhydrous ethanol will absorb whatever remaining water molecules will be there after the gradient. So everything will be removed by the ethanol, and then the ethanol can be dried out using a desiccator. So we don't leave the after doing all these drying steps, all these dehydration steps, we don't leave the sample outside anymore, because in the air we have moisture, right? So that can again, the water molecules can get attracted to the sample, and we don't want any water molecule present in the sample. So what we do, we generally take the, like after doing this anhydrous ethanol step, we take the sample and directly put it in a desiccator where we have the vacuum attached, and under vacuum conditions only the solvent gets evaporated, like it the sample is dried up. Okay, so this is how we do the dehydration step. Any doubt in this? Great, then moving on to the next question, question number 13. Which of the following is a common conductive coating material used in SEM sample preparation? So scanning electron microscopy, as I've mentioned. So which of the following is a common conductive coating material used in the SEM sample preparation? Option A gold, option B silver, option C silicon, and option D titanium. So Agatha answered option A, gold. Okay. Anybody else? Okay, Aditi has also answered option A. So option A is the correct answer. Gold is the most common material that is used for doing the coating. Why do we need to do the coating? Because we need to make the sample conductive. We have, so any metal coating can be done. Generally gold or platinum coating is done. Platinum is more expensive than gold, so gold is more affordable as compared to platinum. We use gold because it is inert in nature, it doesn't interact with our samples, and it will just form a thin film over the samples to provide a conductive surface. So that is the purpose of doing gold coating of the SEM samples. Okay, so all of you answered option A, gold, so you were correct. Yeah, so you can use other, even carbon coating is also done in some cases. So you can use other materials also, but gold coating is the most common uh, coating technique that is used, and it is actually most effective. So silver, gold, platinum, all, if you remember the reactivity series, all these metals come at the very end of the reactivity series. They are very less reactive, so we generally go for these metals for doing the coating, so that it doesn't interact with the sample. We, because we don't want any interaction with the sample, we just want a thin film over the samples, very thin film, like a 5 nanometer or something, and so that our samples will become conductive if they are not. Okay, so that is the only purpose. So gold is the most common material that is used for sputter coating. Why don't we use silicon if we use carbon? That is what I am saying. We can use a lot of materials, but uh, these are not as conductive as gold. So silicon is not as good, like in case of SEM analysis when we are doing SEM. And second thing, sputter coating of gold can be easily done as compared to silicon, because so I don't have the figure for sputter coater, but if you see in your lecture videos, in the lecture slides, there is a uh, diagram of a sputter coater. So you have a piece of gold attached to the attached to one of the electrodes, and then under the vacuum condition, a thin layer from that, like gold dusts are basically sprayed over the sample, you can say, at under the electric field, and this is done under vacuum. So this whole process is not very feasible to be done using silicon. Carbon sputter is also very rarely used, in very special cases it is used. uh, but gold coating is something that is done very commonly for any sample. Like even if you don't know the property of your sample, what you are going to image, the first step is to do like if you don't know if your sample is conductive or not, the first step is to take the sample, fix it, prepare the sample properly, and then gold coat it, and then do the SEM imaging. So that is the most common way. Okay, this is clear. Moving on to the next question, question number 14. What is the purpose of sputter coating in SEM sample preparation? Option A, to add color to the specimen. Option B, to make the specimen conductive. Option C, to preserve the specimen structure. And option D, to remove excess fixative from the specimen. So just now we discussed about coating, so you should be able to answer this. What is the purpose of sputter coating in sample preparation? Sputter coating is the thing that is used for doing the gold coating. So sputter means it's sprayed on the sample. So it's not done continuously. It's just like for 10 seconds we are doing this spraying. So the instrument that is used for doing the gold coating is a sputter coater, uh, which sputters the gold particles on your sample. Okay. So Agata has answered option C. We just now discussed why we were doing gold coating. Why did, in the previous question it was actually written why the gold coating was done. Anyone else wants to answer? Aditi or Rosie, if you want to answer. option B. Rosie says option B. Yes, so that is the reason. So I don't know why Saugata answered option C. In the previous question also I mentioned that gold coating is done to make the sample conductive. If your sample is not conductive on its own, you can add uh, a layer of gold, and gold can give its own electron. It's a conductor, right? So uh, it can emit electrons on its own. So it is helpful. I'll in a minute I'll explain why the sample should be conductive for doing a SEM imaging. But for now, just remember that the gold coating, or the like gold coating is done in the instrument which is called a sputter coater. So sputter coating is done to make the specimens conductive. Okay, I'll explain in a minute why we are doing this. Moving to the next question now, question number 15. Secondary electrons radiated back in the scanning microscope is collected by. The secondary electron radiated back in a scanning microscope is collected by option A, specimen, option B, detector, option C, vacuum chamber, and option D, cathode. Okay, so that says option C, vacuum chamber. Anyone else wants to answer? I'll explain the principle of scanning electron microscope now so that you can answer better. Rosie says option B, detector. Okay. Anyone else? So option B is the correct answer, detector. So this is the ray diagram of the scanning electron microscope. So in the light microscope, if you remember, we had a light source, like source from which a light bulb basically that was illuminating the sample. In case of electron microscope, now replace the light source with electron source. So electron will illuminate the sample. Electrons also have a wavelength, so that they will illuminate the sample now. So electron gun will basically spray electrons, and the electrons will pass uh, through the lenses and reach the specimen. Now this is why I was saying that the specimen should have some conductivity, because when the once the electron hit the specimen, if the specimens don't have any conductivity, the electrons won't bounce back. So they won't have any secondary electrons to emit. So this is the primary electron source. So this is the electron gun. Whatever electrons are coming here are the primary electrons. Okay, after the primary electrons hit the specimen, the they will emit secondary electrons. Okay, and secondary electrons will go in every direction, and the detector will capture these secondary electrons and then transfer it, like it is the electronics part where the image is captured by the electron density, by converting the electron density and all. Okay, so that's how we get an image. So if something has a depth depth in it, so they will show like in that black and white same images you can see the depth and all slightly. So this is the purpose of second, this is the purpose why we need to have the conductive material. So if you have gold coating, it will make the samples conductive, and these secondary electron emission can happen easily, and these secondary electrons will be uh, captured by the detector to uh, see the image. Okay, so this is the principle behind scanning electron microscopy. It looks same, almost similar to the light microscope, but here the lights, instead of the light source, the illumination source is now the electron. This is clear to everyone? Okay, great. Then I'll move to the next question. Question number 16. So this is about TEM now. TEM is transmission electron microscope. Okay. In TEM, how do electrons interact with the specimen? Option A, electrons are absorbed by the specimen. Option B, electrons bounce off the specimen surface. Option C, electrons pass through the specimen forming an image. And option D, electrons scatter randomly inside the specimen. So TEM is transmission electron microscopy. So think uh, of it logically also. The name says transmission electron microscopy. So how do you think the electrons interact with the specimen? Electrons are absorbed by the specimen, or electrons bounce off the specimen surface, or electrons pass through the specimen forming an image, or electrons scatter randomly inside the specimen. What do you think is happening in a transmission electron microscope? Swagata says option C. Okay. Anyone else? Hello. Yeah. Okay, Rosie also says option C. So option C is the correct answer. Electrons pass through the specimen. So why I was emphasizing on the full form of TEM is it in the name itself we have transmission electron. So transmission electron means the electrons are passing through the specimen forming the image. Okay, so in SEM, as you saw, the electrons were bouncing off the specimen, right? In this figure you saw this is for scanning electron microscope, so it is only scanning the surface of the sample, whereas transmission electron microscope can see the inside of the sample, so it can transmit through the specimen. Okay, so this is the principle of transmission electron microscope. So it is again similar to light microscope. This is more similar to light microscope. In light microscope, the source of illumination is a light bulb. Here we have electron gun. The electron gun will spray the electrons, and unlike scanning electron microscope where the electrons hitting the specimen was bouncing back, in this case the electrons will pass through the specimen and reach the detector. Okay, so this is more closer to the light microscope as you saw. So this is transmission electron microscope. So I hope light microscope, scanning electron microscope, and transmission electron microscope, all three concepts are clear to you now. So you have seen the diagram for all three. If you have any form of doubt, you can ask me right now. That would be easier for me to explain. If you don't have a doubt right now, if you want to ask it at the end of the class, that's also fine. Okay. Okay. I think I can move to the next question then. If nobody has any doubt, we can move to the next question. Question number 17. Which of the following are true for electron microscopy? The specimen should be thin and dry. Option B, specimen should be free of dust particles. Option C, electron beam must pass through evacuated chamber. And option D, all of the above. Which of the following are true for electron microscopy? Option A, specimen should be thin and dry. Option B, sample should be free of dust particles. Option C, electron beam must pass through evacuated chamber. And option D, all of the above. What would be the answer? Okay, Rosie says option D, all of the above. Anyone else wants to answer? So option D is the correct answer. You are right. So specimen, first point, that is specimen should be thin and dry. That is true for electron microscopy. Why? Because if the specimen is too thick, the electrons won't be able to just a second, something happened to my screen. I hope my screen is visible, right? So if the specimens are too thick, the electrons won't be able to pass through the sample or even bounce back. They will be absorbed by the sample. So that's why the specimen should be thin and specimen should be dry. I explained this previously also. It has to be free of water molecules, because water or any solvent that is present on the, if something is present on the sample, if the sample is not completely dry, uh, things can get outgassed into the chamber, into the electron chamber, if a vacuum chamber where the electron is present, and it can diffract the electron beam. So in the diagram, as you saw, the electron beams are very focused, right? So if some gas particles are here in this chamber, like in these chambers, if some gas particles are there, they can actually diffract these lights and diffract these electron beams, and they can go here and there, and not all the electron beam that is focused on the sample will reach the sample. Okay, so whatever number of electrons are released from this gun will not reach the sample. Some of the electrons will get diffracted in the way, and we will get a lot of noise because of the diffraction. Okay, so that's why the sample has to be thin, sample has to be dry, and sample should be free of dust particles, because of, again, of this reason, because there are dust particles, you are going to see interference in the imaging. Electron beam must pass through the evacuated chamber. I just now explained, because if there is uh, any form of gas or any form of other particles present, they can diffract the electron beam. So electron has to pass through a vacuum chamber. evacuated chamber means chamber filled with vacuum. So it has no air, no particle, nothing. It's the clean vacuum chamber. Okay. So all the statements are correct. So here you see the electron beam. So if there is any gas particles, instead of going through the lenses, it can actually diffract outwards. So these can actually reach the, probably if there is some diffraction, the diffracted electron beams will also reach the detector, but that those beams are not interacting with your specimen, so they will give a background noise. That's why your sample has to be free of any kind of moisture, any kind of things that can interfere with the electron beam, and the electron has to pass through complete vacuum, because otherwise your beams are going to be diffracted. This is clear? Okay. Moving to the next question now, question number 18. Which type of cells allow entry of trypan blue to assess viability? Option A, only live cells. Option B, only dead cells. Option C, both live and dead cells. And option D, none of the above. Which type of cells allow entry of trypan blue to assess viability? To check the viability, what type of cells allows the entry of trypan blue? Only live cells, only dead cells, both live and dead cells, or none of the above? Rosie says option B, only dead cells. What is trypan blue? Trypan blue is a dye. So, Rosie, you are right. Your option B is the correct answer. So, trypan blue is a dye that can actually uh, enter the dead cells, but it cannot enter the live cells. So, if I'm doing cell culture, okay, so we will have live cells, okay, we will have multiple live cells, and there will be cells that are dying. When the cells are dying, so this is the cell membrane, okay, so when the cell is dying, what will happen? There will be perforation in these membranes, okay, the membrane will get perforated like this. The cell is now dying, it is going to burst. So when we are adding trypan blue, which is a blue colored dye, when we add this trypan blue solution into the cell sample, so we can aliquot like 100 microliter of the culture, and then we can add 100 microliter of trypan blue dye. So what will happen? It will, since these have these perforations, trypan blue can enter the cells, and as a result, here you can see these cells will appear blue in color. So the cells that are appearing blue in color are the dead cells, and the cells that are appearing transparent are the live cells. Okay. So this is how we differentiate between live cells and dead cells, and this is how we assess viability. So what do you mean by viability? Viability means the number of live cells versus total number of cells. So how many live cells are present in the total culture? Okay, in the total culture, so let's say I have 50 ml of culture. Sorry, I have 50 ml of culture, and now I want to find out how many cells are live and how many cells are dead. So I'll take an aliquot out of that 50 ml culture. I'll add trypan blue, and I'll add to the hemocytometer, which looks like this under the microscope. It has these grids formed, and I can actually count the cells. So you can easily count the cells. If your cell concentration is very high, you can dilute it and then add it. So you can count 1, 2, 3, 4, 5, 6. So when you're looking into the microscope, you can count these cells, right? So I can count the number of live cells, I can count the number of dead cells separately. And then there are 4 squares, you can see these bigger squares. Just look at these bigger squares. So there are 4 squares here. 1, 2, 3 and 4, right? So 4 squares. So we'll count the number of live cells and dead cells on each square, and then we'll take the average out, and then we'll multiply with the volume factor, whatever is there for your hemocytometer. So basically, whatever the number of live cells, you will get the average number of live cells divided by the total number of cells. So total number of cells would be number of live cells plus number of dead cells. So this means number of live cells divided by number of live cells plus number of dead cells, right? So that will give us the viability, and then you can, if you multiply it with 100, you will get the percent viability. So how much viable is my sample? So if I get like 90%, that means out of 100 cells, 90 cells are live, 10 cells are dead. So that is what we mean by viability assay. Is this an image? No, it's not an image from EM. It's a light microscopy technique. So you if you see the lecture video, in that there's a demonstration video also, so that will help you understand this better. So we have a slide. Okay, we have a slide where in the middle of the slide we have this grid made. Okay, so this whole thing is made in this region. Okay, and another set of this whole thing is made in this region. Okay, I'll use some other color for you to understand better. So the whole big box, like this whole thing is made here on the slide. Like this grid is already edged on the slide. Okay, and then you can add your sample from this point. You will put a cover slip and then slowly add the sample without like avoiding any air bubble, without adding any air bubble. Then your sample will be here. Your sample has, what does your sample have? So you have your cell culture plus you have added trypan blue. Okay, so now you are adding this sample on the hemocytometer that has this grid made. Now when you look under the microscope at this region, when you look under the microscope at this region, it will look like this. You can see the grid lines very clearly, and you can see the cells, live cells as well as the dead cells. The dead cells will appear blue, the live cells will appear kind of transparent, and you can see like against a bluish background, you can see bright transparent cells, like it will be illuminated by light source, right? So it will be whitish kind of it will look. So you can then count the number of cells, number of live cells, number of dead cells, and calculate the viability. Okay, so this is done under the light microscope. You can actually do it in the inverted microscope also, because uh, mostly in cell culture labs you will find an inverted microscope present. So people generally do it under the inverted microscope only. And what are the central closely spaced lines? Okay, so these lines you mean, right? So this hemocytometer, along with counting cells in a cell culture lab, it is also used for counting blood cells. So these regions we use, like the region that I have circled with red, 1, 2, 3, and 4. These regions we use to count the whole cells. Like these are mammalian cells. So they can be viewed here because they are bigger in size. Now, if I want to count the blood cells, how many RBCs and WBCs present inside the blood cell, now those cells are smaller than the mammalian cell. Okay, mammalian whole cell, red blood cells are smaller. So those cells, when we are when we do blood count, if you have ever gotten a blood test, you might have seen a complete blood count given. So they use these kind of apparatus where you can add the blood sample now here, and your blood cells, the RBCs and WBCs will be seen here. So these are more closely spaced grid lines, so they will have, so these are bigger cells, so we can see them here. If uh, we have very small cells like these, it would get very difficult to count in these bigger grid lines. Like in one grid only so many cells will appear. So that's why we use a smaller grid line, and then the counting calculation and all also slightly differ in that. So this whole thing, it is called hemocytometer, and this is used for cell counting. Depending on the type of cell, that grids are used. Mostly in cell culture technique, these four grids are used that are labeled by red color. Okay. Is that clear? Okay, good. It is also called Neubohr's chamber. So you can find the different names for this on the internet, but the thing is same, principle is same. Okay. Moving to the next question, question number 19. In flow cytometry, what is the primary purpose of the forward scatter channel? Option A, to measure cell density. Option B, to analyze cell size. Option C, to detect small particles. Option D, to sort cells. What is the primary purpose of forward scatter channel in flow cytometry? So now we have moved to next technique, that is flow cytometry. Anybody knows the answer? So flow cytometry is a technique where we can sort the cell. So it's a cell sorter. Okay, it's a cell sorter. So we can sort the cell based on its size, based on its density, and if we have tagged the cells with some fluorophore based on the fluorescence, so we can basically sort the cells. So we can kind of this instrument is able to see single cell, like see each and every cell in a solution. So the solution is made to flow inside a tube, and then there are detectors that can detect single cells. Okay, and those cells can be differentiated based on its size, density, etc. So there are two things, two channels: forward scatter channel and a side scatter channel. So FSC and SSC. Okay, side scatter channel and forward scatter channel. Now, if I have a particle, so it can either scatter the light in an angle, or it can actually reflect it 90 degrees. Okay, so this 90-degree, the perpendicular scattering is basically side scatter channel. The forward scatter channel is the one that goes forward. Basically the scattering is happening for in the forward direction. Now you can see logically also this forward scatter channel has a space between it, This will depend on the size. So if I have a bigger particle, the forward scattering will be larger. If I have a smaller particle, the forward scattering will be smaller. Okay. The side scattering, however, depends on the cell density. Okay. So forward scattering measures the size and side scatter measures the density. So if I have a dense sample, very dense sample, most of the light will be side scattered and we will see very less forward scattering. Whereas if we have a lighter sample, the sample that is uh, not very dense, that will show more forward scattering and less side scattering. Okay. So forward scattering is used to measure the cell size. So option B is the correct answer here. This is clear to everyone? Yeah, once you watch the videos, lecture videos, this will be easier to understand. You watch the lecture videos, then I'll upload the video of today's class also. Then you can again see this video and see the questions also. I'll upload the slides also. So then you will be able to understand better. Okay. So moving to the next question, this is again about flow cytometry. So in flow cytometry, what is the purpose of gating? So I'm assuming since you haven't uh, watched the whole flow cytometry video, it would be difficult. Rosie, do you know the answer? What is the purpose of gating? Option A, to adjust the laser intensity. Option B, to calculate the flow rate of the sheath fluid. Option C, to select a subset of cells for the analysis. And option D, to measure cell size and granularity. So we are running out of time, so I'll just explain quickly. So gating basically, so when, so I told you about forward scatter and side scatter, right? So in a graph, we can, like, once we'll attain the data, we'll attain the data like this only. So let's say we have multiple dots here for different cells. So this will basically show that this region has higher forward scatter and higher side scatter. This region has lower forward scatter and lower side scatter. And this region has medium. So now let's say I am interested in looking in this region. So what I'll do gating. Gating basically means I'll look in this subset only now. So I'll just ask my instrument to look into this subset, take the readings in this subset so that I can get a better idea of what is there in the mixture where we are getting high side scattering and high forward scattering. Okay. So basically to select a subset of cells, so there are different ways of gating. I'm not going into the detail that you can easily get from the lecture videos. So this is the purpose of gating, to select a subset of the cells. Now you can select it like a box, you can select it like a region, like if you divide the whole thing in quadrant and then I can say show me the uh, region, or first region, second region, third region, or fourth region. So that depends on how you are doing the experiment and what your instrument allows. So but this is what gating means, because you want to select the region where you want to look further into. Okay, so you do first set of analysis, you see the graph, and then you select a particular region because you think that, okay, this is the region I want to focus on. So you will gate that region and you will ask the instrument that show me more data about that region. That is how things work. Okay, like I'm explaining it in very simple language as possible. Like when you watch the video, you will understand the technical term of it. So option C, that is to select a subset of the cells for analysis, is the correct answer. So moving to the next question, question number 21. Compensation is performed in flow cytometry because of dash. So this is the last question. Compensation is performed in flow cytometry because of dash. Option A, fret between fluorophores. Option B, low intensity of signal. Option C, spectral overlap between the two fluorophores, and option D, poor sample preparation. So compensation is again a technique. I'll directly explain this to you. So the correct answer is spectral overlap between the two fluorophores. So uh, now when we are doing flow cytometry, we can actually tag the different cells with different fluorophores. Okay, let's say I tagged one type of cell with red fluorophore and another type of sample with green fluorophore. Now when we are doing this experiment, there is a chance that we get an overlap between the two fluorophore signal. So when we are getting the data, so let's say we get the data for one fluorophore signal here, and we got the second fluorophore signal overlapping with the first fluorophore signal. So this region, with blue, so this region that we have, this is basically an overlap region. So this will give us a false data, because the cells that we see in this region could be a part of the first fluorophore or could be a part of second fluorophore. We won't be able to differentiate. So then what we do, if this is our baseline, what we do, we kind of, like, our instrument will tell us that how much contamination is there from one fluorophore into the other, and then we can attenuate this so that our baseline is somewhere here. So if I shift my baseline from here to here, now I will get a graph clearly separating two peaks, right? Because here the peaks are separated, and this region, if you see, the peaks are separated, right? So if I now shift my baseline here, my peaks will be well separated, and I can see, okay, these many cells are there. And so this is basically compensating. So when we are doing this, when we are shifting the baseline, so we are losing the data of these regions as well, right? So this is kind of a compensation that we do in the flow cytometry data. So to compensation is done to because of the spectral overlap between two fluorophores. Okay, so that is the last question of this session. Anyone has any doubt in the whole, any of the topics? Microscopy, flow cytometry might sound a little trickier, but I would fluorophores are the tags that can fluoresce. So something that has fluorescence on its own. So there are different chemicals that have intrinsic fluorescence. Okay, so there are there is a chemical called FITC. Okay, so it's a chemical molecule, organic molecule, and it has a fluorescence. Now that molecule can be attached to any cell. So this is called a fluorophore, because now we are using this molecule as a tag. So it is, and this tag has fluorescence, it is called a fluorophore. Okay, so it's like a label. Okay, so any fluorescent label is called a fluorophore. So I would suggest go and watch the lecture video of flow cytometry, and then watch the recording of this meeting also. Again, you will find the recording in the problem solving session. I'll upload the slides also. Then you read the question again, try to understand the lecture. it would be easier. Okay. So, any other doubt? Okay. Then I think we can end this meeting now. So, thank you all for joining. I'm